Thank you, Kevin and Daria, for inviting me. It's such a great pleasure to be here at Stanford again. One of my true translational and transitional points in my life. Before coming to Stanford, I was very much interested in the development of the brain and how synapses formed. And coming to Stanford introduced me to for the first time in my career to the concepts of translation and of clinically relevant problems, something I'd always thought about. And it was actually Dr. William Mobley who actually introduced me to Down syndrome and said, can you begin thinking about all the things that you think about at synapses and how that might apply to Down syndrome? And so that was the beginning. And the next really major step was a discovery, which I'll tell you about. And thank God for Kevin and Daria, because in the year where we're really trying to figure out what to do next, where we'd made our discovery, they provide us a solution of how to move forward. So I'd like to tell you about that. So this story begins effectively with this four gentlemen. Like all the projects that have been really great in my life, it's been a group of people that have been really spectacular. The story begins actually with Fabian Fernandez, a graduate student in the laboratory who started working on Down syndrome in my laboratory. And he was the genesis and the genius behind the therapy that we ended up developing. And Fabian was really a remarkable young man, and his story began because he, looking at the field of Down syndrome, recognized that there were no solutions available. And he synthesized lots of research both in clinical and basic science, and came up with a really novel idea, which I'm going to tell you about. And the two other gentlemen, Dan Wetmore and Craig Heller, here at Stanford, were inspirational in helping us at every step of the game. So the problem is kind of simple and complex at the same time. So Down syndrome, as you know, is a neurodevelopmental disorder. It involves the triplication of an entire chromosome. So for those of you who are challenged by a single gene increase, this one is the mother of all. There are over 300 genes that are triplicated in these children. It's, as you know, children do quite well with this. They have a variety of different disorders, including growth and leukemias, etc. But we were most interested in the one related to cognition, since this is what really holds them back. There are many things that are affected, but in terms of cognition, they can have anywhere to be severely impaired with an IQ of 20 up to an average around 80. Interestingly, the defect is really largely centered around acquisition and storage of knowledge. And this inability to do this impairs their ability to learn language and other kind of rudimentary things that we take for granted every day. It also has an unfortunate secondary problem, and that is that every child with Down syndrome by the age of 30 to 35 will have the pathology of Alzheimer's disease. So this contributes in many ways. So in actually trying to develop a therapy for the first, we are actually thinking a lot about the second. So up until the 60s, the standard of care was institutionalized. And of course, this didn't help. And one of the best things that happened to this population was inclusion education, which brought children with Down syndrome home. And this was probably the most transformative for this population in terms of being able to now acquire information. But still, this was not sufficient. And there was no other treatment for them. They would give them vitamins, vitamin C, a lot of different things were tried, but none of them worked. And so we were considering what you could do to do more. So I already mentioned Fabian, and his central hypothesis from a lot of work was that the circuits within the brain were not working properly, in part because the inhibitory ones, the ones that actually prevent change during learning, once you've acquired information, that these were elevated. And this illustrates it here. This is kind of a chart which gives you the performance of a network and how well it works. Okay. This tells you performance, so this is where most of us sit. We have an optimally performing brain. But if you look at, in a very simple way, the excitation inhibition balance in the brain, if you are on this side over here with too much excitation, you have seizures and epilepsy. And on this side, you are in a coma. So those are two extremes. And of course, those of us who experience opioids or alcohol, know that you then are somewhere on this side of the curve, and your performance is drastically reduced, and you would effectively behave a lot like a child with Down syndrome if you were on a benzo. So this was basically his idea. He looked at the literature and basically concluded that there was too much inhibition in the brain. And therefore, after looking at a lot of things, he said, if that's true, there should be a simple solution. And his simple solution was, what if we were to give a benzo, was that if you would take away this inhibition, you could drive the circuit back to normal. So he predicted that they were sitting somewhere here, and if you simply gave a GABA-A receptor antagonist, that you could return the circuit to normal function. So we had a mouse model in which there was a triplication of most of the genes in Down syndrome, and they had the same kind of behavioral problems in hippocampal learning deficits. And that's just illustrated here, where we're doing an object recognition task where you give them 2 objects today and then give them 2 new objects tomorrow. And like all animals that like novelty, they readily spend more time with the novel object. And this just illustrates that these mice do not learn. They see both objects equally well. So after giving them a 2-week regimen of like 5 different GABA-A receptor antagonists, he got the following. And that was, if you gave one of these drugs for 2 or 3 weeks, and then you waited a week, you saw that the performance was back to normal. What was even more remarkable was that 2 months post-treatment, you were still at normal levels. So this imprinting, this kind of repeated dosing over a 2-week period of time, completely allowed the circuit to now operate in a better way. So this then opened up the idea that maybe we had a treatment that we could actually develop for people. So here came SPARK. We were totally lost. We're trying to figure out what we were going to do. We're like most academics. We did the science the best we could. And so what we really needed is a mechanism to actually take the idea and move it forward. And these are the things that SPARK did for us. So first of all, through their team of advisors, they basically honed us in on the most important. We had 8 different drugs that could be useful, and they said, pick this one, it's been in man before. And then they said, this is the list of things that you have to do. If you're going to imagine using a GABA-A receptor antagonist, this will lead to seizure. So we need to know, what is the efficacious window? You know, where is it going to be efficacious and where is it going to be dangerous? And if you can figure this out, we think you can have a winner. And so that's exactly what we were able to do. We actually found there was a 1,000-fold window of opportunity for this drug, and so that was a very exciting thing. They also provided certain guidance in terms of dealing with our patent attorneys for getting a use patent. They introduced us to angels and venture capitalists, and perhaps the most important piece was they introduced me to Lyndon Lin, the CVO of the company that I then founded. Because in part what I realized that I was a pretty good inventor, but I was not a businessman, and to find an expert like Lyndon who had 25 years in pharma was really a godsend. So we founded a company, Dan Wetmore, myself, and Lyndon, called Balance Therapeutics. And we took this initial idea and we did raise money and did a phase 2 clinical trial. We did all the phase 1 work again, did a phase 2 trial in Australia, which finished this past year. And it was actually a success in the sense that in the phase 2, we see an improvement in the cognitive performance of them. It didn't cure all the things that they have, so this will be a challenge going forward, but at least we're one step along the road towards developing a cure for these, or a treatment for these children. In particular, it tells you that this is one of the neural network systems that is not functioning well, and that if you take a similar strategy, you might be able to do better. Now, as a group, we're thinking about this more. So here's this curve again, and you can see all these different indications which sit on the overexcitation and the overinhibition side. So conceptually, we think that our treatment with this GABA-A receptor antagonist, that we might be able to move several of these indications forward towards an improvement. And so we just initiated a series of 3 new clinical trials in the ARISE. The 2 listed here are for idiopathic hypersomnia and also narcolepsy, which both of which are caused by increased inhibitory tone in the brain. And so these trials are just getting underway. So with that, I would like to say it's been a fantastic experience being part of SPARK. I'm using it now to run a SPARK program in Berlin, which is tremendously exciting for me. And it's really about the kids and the children that we're trying to help. And with that, I'll stop. And thank you very much for your attention.
So we have just a few minutes for questions, if anybody has any.
We are developing similar drugs, so I'm interested in very much. So it's very difficult to try clinical trial tests in children. So how to persuade the government, Australian government, to do the clinical trial? That's my question.
So I think it's, in this particular case, it wasn't, it was partly government, meaning we had to work with the FDA. So we were very strategic about the drug that we were using. It was one that had been in people since 1920, although it was no longer approved. And so we were able to actually capitalize on that and use the long history in man before that time. And so this was what helped build our case for the FDA to eventually approve that we could go forward and test it again. In terms of the children situation, this is always a bit complicated. In this particular trial, we began with everyone, only people older than, I think I had it listed here, older than 13 years of age. So again, going into yet younger, which eventually one would like to do because that would be important. This was one of our way out. What was important also, I didn't have time to talk about, is we found that it didn't matter when you give the drug. It didn't have to be in the first weeks of life. You could do it in an old or an aged animal, and it was equally useful, and similarly useful in Alzheimer's animals. So those that have this higher inhibitory tone, if you reduce it, the circuits will begin functioning again, at least for a while.
Sometimes, parents of the Down syndrome children are very reluctant to cure their symptom because they insist, Down syndrome is not a disease, but the character of the person. So, yeah, we have such kind of trouble. So
this is always a problem. I think it's like all populations. There are some families within this that are very progressive and want to see new treatments come. And I think the others, it's a difficult disorder for which one learns to accept and move on. So I think if we can provide something that would really work, this would begin to change people's opinion. But I do understand where they're coming from.
Okay. One last question.
So can you share with us some of your experience in translational work? How do you use animal data to translate to human, like when you develop this kind of a balance between inhibitory and stimulative neurotransmitter balance?
Yeah. So animals are just that, a model. And I think what they allow you to do is learn something very fundamental about mechanism. We know that they're very challenging in terms of direct translation. You cannot really use them in a perfect way because they're not exactly the same. It's also, I think it's important to pay attention to the measures that you're going to use. So some measures are more applicable than others. This particular one for Down syndrome is a very hard one. It's probably the worst one that you could possibly pick because it's dealing with cognition, which is a very small change. So even if you saw a 15% change, this could be quite big in a human over time. So while in the animals it seemed to improve them completely, in our studies it's a much smaller percentage. So I think one has to use animals as more of an understanding than a perfect tool.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.